quiz, maybe two more before Tuesday. Instead of thinking of it as a burden, just think of it as a way to get you working on the material before Tuesday. So, I'd like to do a quiz, probably not last stuff. Maybe just one quiz. That'll bring us to 7 quizzes. Then if we do 5 more quizzes, 12, I would drop 2, right? And your highest 10 would count for it. Or maybe we'll do 4. I don't know. We'll see how. By the way, I want to make sure we have more than 10. 12 would be better. So just in your mind, be thinking about maybe Sunday again. I don't think there's a class in here before us. Has anyone been sitting in here like, I don't know, since 11:45 and can tell me? Is there no class? So if you want, okay, spread the word. I'm going to show up here 11:45 on Tuesday. So if you want more time to take the exam, it's probably your interest to show up at 11:45. If you don't, I don't care, that's fine, show up at 12. Like, some people are really quick, some people want more time, that's cool, but I'm going to try and give you a little more time. However, the exam will be similar format, should take a similar amount of time, see what I'm saying? So you get 15 minutes more, should help you. I expect some drawing questions like before. I'm still working on the study guide. I promise I'll get that out to you by end of today, before midnight. For example, you should probably be able to draw a simple reflex, like a stretch reflex, which I'm going to do again right now, and understand the basic parts, maybe a neuromuscular junction, you know what I mean, and maybe some basic retinal circuitry. And in my study guide, I'll give you a sense of some of the things I hope you can draw. Do you know what I mean? And again, the purpose of drawings isn't to draw like Leonardo da Vinci or something. It's just a great way to synthesize and integrate information. And there's not one way to do it, okay? So I don't grade you on how beautiful your drawing is or if it's a certain template. It's just, I find it a really efficient way to convey a lot of information. I don't like reading essays. I'm a bad writer. Most of us are. And so I don't want to read that. So instead, you can give me that information in a picture. It's easier for me to grade. And it's still information dense. That's kind of why I like to do on exams. And maybe it's a little different from some of your other exams. I don't know, maybe some other props that you draw. I don't know. So, that's what I'd be thinking about if you're trying to think about how to prepare for Tuesday, right? And like on the first one, I just go off what I did in here in my PowerPoint. If I understand if some of you, imagine you're not, because you're all busy and a lot of you are working too. Parents, like I know we're all stressed and we've got a lot on our minds. But some people maybe wish there was a textbook. I purposely didn't give you one because I'm saving you money, but also, in my experience, people don't read them. But if you want, I'm totally happy for any particular topic. I can try and point you in a direction towards resources that I think are cool or interesting, or like review articles. That's usually where I go. So like, I'm happy to point you in that direction if you wanted more than just what I'm giving you, which is just my take, my little feeble take. So that's logistics. Make sense? And then next Thursday, we're just getting right back into things. Although that's going to be on Zoom. Last time on Zoom, right? But we will start the next unit, which will actually be a continuation of motor control studies. We'll do that for a few more days. And then we're going to get on to the cerebral cortex. Actually, we've already started thinking about cortex, haven't we? We're going to continue talking about cortex. Other cool stuff you'll find interesting. So, let me review really quickly what we talked about Tuesday. Arguably the most important tract in the nervous system is the cortical spinal tract, sometimes abbreviated CST. And it starts in the cortex. Here's a little human brain, and this is a coronal section. So here's two hemispheres. Here's a left and here's a right hemisphere. I really should reverse that, because I really should say this is right and that's left, because on radiographs everything's reversed, right? You guys do healthcare. You gotta get used to looking at things reversed. Anyways, make it easier. So, two hemispheres of the brain, and coming from multiple, let's just take the left hemisphere, frontal lobe, multiple regions. You have this motor tract, and it makes its way down through the brain, and now we're going through the brainstem. I really should put a diencephalon here. Diencephalon, brainstem, and we get to the bottom of the brainstem. And we're learning a little bit of anatomy in this course, so there's some things I want you to know. Remember, there's three parts to the brainstem: midbrain, pons, medulla, and then right at the bottom of the medulla, this tract crosses to the opposite side. And now we're going down the spinal cord. The place it crosses is called the pyramidal decussation. And it goes to really all levels of the spinal cord. Let's just take a random level right here. And then into the ventral horn of the spinal cord. I'll show you a cross section too. I'm going to show you a cross section right here in a second. What's interesting is most of these fibers actually don't directly innervate a motor neuron, which I think they would do. They innervate an interneuron, and then that interneuron innervates a motor neuron, and that goes to skeletal muscle. And I finished Tuesday, well, right before I started talking about reflexes, I spent a lot of time talking about the neuromuscular junction. So that's this synapse here, right, made between a motor neuron and skeletal muscle. That's called the neuromuscular junction. Okay, anywho, so that means, right, left side of the brain controls right side of the body. That means if you have a left-sided stroke, what do you see? Right-sided weakness. People's limbs will sometimes end up kind of flexed, pronated, and symptoms on the opposite side, stroke. You follow me? So that's the cortical spinal tract, and we call it an upper motor neuron tract because it's in the CNS. And we call this guy right here, this alpha motor neuron, whose cell body is in the spinal cord, but then goes to skeletal muscle. We call it a lower motor neuron. I'm doing a little bit of review. I'm going to stop here a second. And I talked on Tuesday about it's kind of interesting, maybe unexpected for you. If a lower motor neuron is damaged, we see one set of symptoms. If an upper motor neuron is damaged, we see a different set of symptoms. The similarity with both is you get weakness and paralysis. But interestingly, if you damage lower motor neurons, like in polio or neuropathy, like in diabetes, you lose reflexes. And how like doctors have the patellar ligament and you kick your leg, okay? Those reflexes are lost. Contrast, if you have an upper motor neuron syndrome, like following a stroke, where the cortical spinal tract is damaged, you test reflexes. They're explosive, they're hyper-reflexic, and they resist, not consciously, but when you try and move their limbs, even though they're paralyzed, it's like lead, okay? We call that spasticity. Whereas in lower motor neuron disease, it's floppy. You can easily move the limb, okay? So we talked about a distinction between upper and lower motor neurons. Does that make sense? Big picture, this is how I remember it. Upper motor neuron tracts, like the cortical spinal tract, are in the CNS, and lower motor neuron tracts, like alpha motor neurons, are in the PNS. Easy. Okay. So here's a cross section of the spinal cord. I said I would do it. So we were taking a cross section of the spinal cord, and you know it has gray matter that looks like a butterfly. If you're in the thoracic region, there's a little horn here. So these are called dorsal horns, lateral horn, ventral horn, and so this is a little central canal. So this butterfly-shaped region inside is gray matter, and then all the rest of this stuff is white matter. So that's actually opposite to how the brain is organized, right? In the brain, it's gray matter on the outside, white matter is internal. Okay, so let's now get back to the end of the lecture, and we're just going to talk a little bit more about reflexes. So if I tap the patellar ligament, even though it's subtle, tapping that ligament, I'm actually stretching quadriceps a little bit. And that stretch is sensed by a proprioceptor called a muscle spindle, okay? And so if I tap quadriceps, the patellar ligament with my little reflex hammer, that's horrible. But anyways, quadriceps, okay. So we talked about how most of the mass of a muscle that you see is extrafusal muscle fibers. Extrafusal. Okay, but there's like a little subset in there called intrafusal. And wrapping around these is a sensory proprioceptor, and it's got a cell body and a dorsal root ganglion. I want you to know where these cell bodies are, okay? Like motor neuron cell bodies are in the spinal cord. This sensory neuron cell body is in the dorsal root ganglion. We're outside of the spinal cord, if you follow me. And it just got activated because when I tapped this ligament, the patellar ligament, I stretched quadriceps, and that stretched these muscle fibers, and that triggered activity in the sensory receptor, okay? We call this a 1A proprioceptor. So it's gonna come in to the spinal cord, and I'll just do a really simple version of what happens. And synapses on a motor neuron going to the same muscle that was just stretched, quadriceps. See it? And the cell body of this motor neuron is in the ventral horn. See how I'm emphasizing where the cell body is located. The sensory one is out here in a ganglion in the peripheral nervous system. The motor neuron is in the spinal cord. You follow me? And this guy now is active, and it just goes right to the extrafusal muscle fibers. There's a little neuromuscular junction there, and it contracts quadriceps. So that's the reflex. I tap patellar ligament, I stretch quadriceps, and quadriceps contracts. Okay, we call that a deep tendon reflex. It's also called a stretch reflex, or like a myotactic reflex, whatever. And just so you know, let me just repeat this. If you have upper motor neuron damage, reflexes go up. And if you have lower motor neuron damage, reflexes go down. Yeah, I mentioned this earlier in the course, right? We have this intuition that if you're paralyzed, no movement is possible. And that's wrong, right? It just means you can't consciously move. Your limbs can still move in based on activation and reflexive circuitry. See what I'm saying? So like, you could have paralysis of your right lower limb and you can't move it, but if you tap the patella ligament, you're going to see movement of the right lower limb. So there is movement possible. It's just that you can't consciously do it. See the distinction? Okay, so that's a stretch reflex. Now, we could make the circuit more complicated because you know how across a joint we have agonist and antagonist muscles, for example. Biceps at the elbow flexes, triceps extends, right? Quadriceps extends the leg at the knee, my hamstrings flex. Got it? So it kind of would make sense if you're trying to contract quadriceps, maybe you want to inhibit the hamstrings, otherwise like they're both acting at the same time, and that doesn't make sense. So we could make this reflex circuitry a little more complicated. This is what we see. You could have a little fiber innervating an interneuron, and remember most interneurons are inhibitory. And does anyone remember the neurotransmitter that inhibitory neurons use in the spinal cord? Glycine. And so this guy's going to inhibit a motor neuron going to the antagonistic muscle to quadriceps, which is the hamstring. So I'm just going to put the hamstring here. So we can make the stretch reflex more complicated. You see what I'm saying? So the same sensory neuron that was activated by stretching the quadriceps when I tapped the patella ligament, it's going to innervate a motor neuron directly excited to quadriceps contraction, and via this inhibitory interneuron using glycine, we're going to inhibit the hamstrings. Mission accomplished. Make sense? Okay, that's stretch reflex. I'm just doing the end of today's lecture bed on the whiteboard. But okay, let's do a different example. So here's a cross section of the spinal cord. And now I want to talk about a different kind of reflex, which is a protective reflex. Those stretch reflexes, I mean, the question might have arisen in your mind, like, why do we have them? Because, like, we didn't evolve to have a ligament tapped by reflex hammers. So, like, what's the point? They're just involved in balance and things like that. And adjusting posture, kind of going on underneath the surface of our consciousness, which is what reflexes are, right? But we have protective reflexes. I've used this example before. You're talking to someone in the kitchen and it's such an animated conversation, you forget that the tea kettle's hot. It's happened to me, you just burn your hand. But luckily, you don't have to consciously like register, oh God, the tea kettle's hot, I better move my hand away. It just happens. We call that a flexor withdrawal reflex. It'll just happen naturally. Okay? Or in your lower limb, if you step on a shard of glass, your foot will just reflexively flex away from the shard of glass. So let's do that now. So now, the sensory input is not a proprioceptor like it was here, right? This 1A fiber is a proprioceptor. I should have said this. And this like little linkage between the sensory receptor and intrafusal fibers, we call that a muscle spindle, right? So that's a proprioceptive organ. And so that was the trigger for that reflex. But what I'm talking about now, the trigger is a flame that your finger is touching. Ouch. Okay, so we're gonna flex our limb away from that flame. Not consciously. This is reflexive. So what's the neuron that got activated here? It's a pain receptor. Where is its cell body? Also in a dorsary ganglion. Okay. So, little pain receptor. what do we call those? Nociceptors? Nociceptors. You could also activate thermoreceptors, we talked about that. And this guy is going to come in here and synapse on a motor neuron that goes to flexor muscles to, pull your limb away. And, if it was your finger, it would make sense to have this motor neuron flex, for example, biceps. Biceps would flex, right? Your forearm. And your fingers would move away from the flame. Make sense? Okay. There too, you clearly, if you got to flex this limb away from a fire, you don't want triceps pushing your limb back into the fire. Okay? So just like in that other example, it would make sense to also activate an inhibitory interneuron, which is releasing glycine again, and suppress, therefore, activity to extensors. Okay? So net result is we burn our finger, we activate flexors to biceps, we flex our limb away from the flame. We also want to inhibit triceps, otherwise what would happen? Triceps would extend our hand right into the flame. That would be a stupid reflex. So, we have kind of cool, complicated circuitry just in the spinal cord. But now, let's do a third example. These are fun. Okay. Third example is for the lower limb. Because for the upper limb, if your right hand's being burned, it kind of doesn't really matter what's happening to your left limb. Like if I flex my right limb away from the flame, it's not really causing much imbalance to the other side of my trunk. You follow me? But imagine this scenario. I'm walking barefoot in the kitchen. Have you had this happen to you? And you step on a shard of glass. You're also going to trigger a flexor withdrawal reflex, right? So I'm going to flex my limb away from the glass. But what could happen? Now I'm out of balance. I could fall over, hit my head. I just got a cerebral hemorrhage and I died. Thank you, flexor reflex. Bad. So what I want is a reflex that simultaneously flexes my right limb away from the shard of glass and activates extensors in the opposite leg. See it? My extensors are activated here, so I'm like firmly planted on the ground. So when I flex my limb away from the shard of glass, I don't just fall over and get brain damage. So that's called a crossed extensor reflex. And so here's the spinal cord cross section. Dorsal horn, ventral horn, and now we're thinking about the lower limb here. So we just stepped on a shard of glass, and so we activated a nociceptor in our foot. The glass was like the trigger. Cell bodies in the DRG again. This information comes into the spinal cord, and we activate flexors over here in the lower limb that flex our leg away from the glass. But we think, it could be, there's different variations of this. You'll see different pictures drawn because the circuitry that I'm giving you is just like a simplistic version. But we think this sensory fiber coming in, part of it crosses and activates sensors on the opposite side. So one sensory input activates flexors on the side of the insulting glass, or whatever you want to call it. And then on the other side, we activate extensors so we don't fall over. See it? So it's a cross sensor reflex. And again, you could imagine this is more complicated, right? Maybe on this side, you don't want to also activate extensors. Then you'd be driving your foot into the shard of glass. So we want to activate, again, like an inhibitory neuron. And we're going to inhibit extensors over here. Okay, you get the idea. So, some simple reflexes in the spinal cord. But, okay. That's that sometimes makes people think, well, sometimes people just think that's boring. Like, okay, great. You know, there's 180 billion neurons in the brain, and you know, like all you're talking about is a simple stretch reflex in the spinal cord. Who cares? So that's just the simplest reflexive activity that the spinal cord generates. If you, did I talk about essential pattern generators? Maybe a teeny bit, but it's okay. Repetition helps, right? But I always forget to never talk about it in the class. I want to come back. These are pictures of these reflexes. But so did I talk about this? I think I did. So this is an experiment done in cats. And then I'll talk about some human relevant related experimental data. So if you take a cat, it's kind of, you could argue it's kind of a cruel experiment, but it's done in the name of science. But anyways, if you separate the brainstem from the spinal cord, okay, what did you just produce? You produced a quadriplegic cat. It can't move any of its limbs anymore. It'll survive, okay? Just like if you're in an automobile accident and you damage cervical cord, you'll become a quadriplegic. You can't move either, but you'll survive. Who was that? Who played Superman a long time ago, and Christopher Reeves, right? He ended up like in a horse accident and ended up quadriplegic the rest of his life. So, back to the cat. So now we have a quadriplegic cat. It can't move, right? Right. Now, if we put it on a treadmill, let me turn this. Okay. And, you know, a little more details I'm leaving out. But anyways, you put it on the treadmill, you kind of suspend the cat on the treadmill so its paws are just on the treadmill. And you turn the treadmill on, like at a gym. The cat will start walking. Flexion. Front limbs, back limbs. You can see alternating flexion, extension. It'll even generate different kinds of locomotion behavior. Walking, trotting, pacing, galloping. This is a paralyzed cat. It's not even just that one limb is paralyzed, all of them are. You just cut completely through the spinal cord. There's no way its brain can talk to its spinal cord. And yet it's exhibiting locomotion behavior. So the only way that locomotion behavior could happen is if there's really complex reflex circuitry in the spinal cord doing it. So as much as you might think, that's boring, stretch reflexes and withdrawal reflexes, you just drew for us 2 or 3 neuron circuits, boring. Actually, no, that's not all the spinal cord can do. It generates locomotion behavior. In insects that fly, flying behavior, that's also controlled by pattern generators in the insect equivalent of the spinal cord, okay? Swimming behavior, that's also generated by complex reflex behavior in the equivalent of the spinal cord. So we call the neural circuitry that generates really complex movements like flying, swimming, walking, running, galloping, jumping, central pattern generators. So, so it's a mistake to think that the spinal cord just does this really kind of stupid flex my limb away, reflexive activity. It's actually generating really complex stuff. Now it's true. Clearly, we can choose to walk and change the pace that we walk, and change the direction that we walk, and decide to run, and then decide to jump. So clearly, these descending motor tracts that we started thinking about Tuesday, like the cortical spinal tract and this brainstem motor tract, clearly they can talk to the spinal cord, influence what central pattern generators are doing, right? But you can separate the brain from the spinal cord and still get walking and running and swimming. So that's my point with this cat example, which is pretty wild, right? Pretty cool. And so the other thing is, if you read the text here, the other interesting thing about these central pattern generators is, unlike those reflexes I just described Tuesday, I just drew on the board. Remember all those reflexes: stretch reflex, withdrawal reflex, cross extensor reflex. It all required a sensory stimulus, right? What was the sensory stimulus for a deep tendon reflex? Stretching a muscle. What was the stimulus for a flexor withdrawal reflex? Some noxious stimulus like a flame or a sharp piece of glass. Central pattern generators, that's just a circuit built into the spinal cord that will generate walking. You don't need sensory input for it to work. So that's an interesting distinction. And this is just a cartoon representation of a kind of experiment that people do with different species. This is an insect, and if you go back to our first unit, right, and you think about insect nervous systems, and they have these long sets of ganglia connected by longitudinal nerve fibers, and the little fibers going out to the limbs. And if you record from these, you can see alternating activity in elevator and depressor muscles for the wings. See what I'm saying? That's just, you don't need, remember we talked about like cephalization as evolution, as nervous systems evolved, okay? You don't need that. You just need these little ganglia here in a line, which is like the equivalent or analog to our spinal cord. You see what I'm saying? So we see central pattern generators in insects. This is an example of medulla, human brainstem. You're wondering, what is that thing? This is a cross section through the brainstem. These little numbers represent cranial nerve nuclei, like that's the vagus nerve, if you wanted to know, and that's the nerve that goes to your tongue, if you wanted to know. Anywho, these are the pyramids that we talked about Tuesday. Our cortical spinal tract is so big, so many fibers. When it gets to the bottom of the brainstem, it starts to bulge out. And one of the cool things about the medulla is it's got this circuitry that controls respiration and cardiac rate with, some help from other brainstem regions. So in a way, that's kind of like central pattern generator like too. So it's not just the spinal cord, but we also see similar kind of neural circuits in the brainstem, okay? And you can see in patients with brainstem lesions and disease of one form or another, they'll start to get alterations in their respiratory rates. You'll see interesting changes in respiration. So like the central pattern generators are getting tweaked, okay? Some of this work has been done in crawfish and in lobsters, and again, I'm not going to ask you to draw one of these central pattern generators, but you can see it's pretty complex circuitry that we've mapped out. Just what are they doing? They're doing electrophysiological recordings from neurons in this chain of ganglia running in this animal, and you can see, rhythmic kind of activity going on here, right? And then people are thinking about, well, what's generating that rhythmic activity in these central pattern generators? If it's not like the brain talking to it, forcing a rhythm on it, these neurons themselves must be capable of, intrinsic rhythmic activity. And so you see neurons that have really cool like bursting patterns, and some that have like this little plateau potential, lots of spikes there. So anyways, and then you can see neurons, groups of neurons have coupled behavior. See this? And you can start to see how you could build a really complex circuit that could generate alternating flexion and extension movement like in walking, or compression, elevation like in flying, okay? So just examples of the kind of research that's done looking at the neuronal properties in these central pattern generators. Kind of cool, right? Okay, so that's the end of that. I could go back if you want me to these pictures, but I think I drew it on the board. I'd rather just, like this. You want me to go over? Maybe a faster way to do this. You want me to go over these reflexes again? I kind of did it. You do? So only one person saying yes. 2 people are okay. So, so here, we can see. The sensory input, right? These 1A fibers linked to spindles, and we can see they're talking to motor neurons going to the same muscle that was just stretched. That's a simple stretch reflex. But we can also see, right? Some fibers are talking to inhibitory neurons, and they're going to inhibit the antagonist muscles. Okay? So we see the beginning of complex circuitry here, right? Central pattern generator circuitry being like the extreme example. And here's just a little look inside. What are we looking at here? This is a half of a cross section of the spinal cord, right? And this is that gray matter that looks like a butterfly. We did both sides. And they're just showing you the kind of electrophysiological experiments you can do where you're recording like from these 1A sensory neurons, and we see action potentials in these guys. So they're sending that stretch information into the spinal cord. And if we record, from this extensor muscle here, we're getting a little bit of positive activity there, but now we're inhibiting the antagonistic muscle here. And because of this inhibitory interneuron, we actually see, hyperpolarization in that flexor. Am I making sense? So we see excitation in the extensor of this reflexive circuitry, and we see inhibition in the flexor of this reflexive circuitry. This is a reflex where the extensor was activated. Here's a case. This is a real-world case where you can see, why a stretch reflex might be helpful. Say you reach and grab a mug of beer, and it's a pretty big mug of beer, and so there's a little weight to it, right? That weight is actually going to stretch initially your biceps. Okay? And if it was a really heavy weight, it might stretch it so much that you drop the cup. It kind of makes sense. You trigger a reflex, that little stretch, to contract biceps a bit, right, to counteract that stretch. Now we can hold that object and won't fall. You see it? See what I'm saying? So it's kind of a real-world example. Or if you're holding an empty cup and you ask someone to pour liquid into it, again, as you're pouring liquid into it, it's adding some weight, it's stretching biceps, and you don't want that cup to fall, so that reflex is gonna contract biceps, counteract that stretch. Okay? This is just showing you most of the muscle fibers in our skeletal muscle we call extrafusal. And we have this little subset of cute little intrafusal muscle fibers. And these guys are linked to those 1A sensory neurons. And that whole thing, we're going to call a muscle spindle. It's a kind of proprioceptor. All right? And, okay. Yeah, I guess there's one more piece of information. here's a question that arises. If muscle spindles, looking for a prop. If muscle spindles are really good at sensing stretch, okay, then they're really good at telling the nervous system when a muscle is stretching, when its length is increasing. But think about it. When a muscle contracts, it shortens, right, typically. So that's like half of the time what muscles are doing. So does that mean that half of the time muscle spindles can't tell central nervous system what the muscle, are you following me, what the muscles are doing? If muscle spindles sense stretch, increase in length, then they probably are silent when a muscle shortens. That means the central nervous system is only getting half of the information about what our muscles are doing. That's not good. The cerebellum needs to know always, are my muscles stretched? Are they shortening? Just to create smooth, coordinated movement. You following me? So how do we get around this problem? Turns out there's a second kind of motor neuron coming from the spinal cord. It's called a gamma motor neuron. Remember, alpha motor neurons whose cell bodies are located in the ventral horn of the spinal cord. These are the guys forming a neuromuscular junction, contracting skeletal muscle. Cool? All right. What I'm saying is, if we stretch a muscle spindles are really good at informing the CNS. That just happened. But if a muscle shortens when we contract it, muscle spindles, if that was the whole story, they would be silent and the CNS would not be informed that, hey, my biceps just shortened. Okay? Cerebellum then couldn't do its work. It's got to be another story. So it turns out there's a second kind of motor neuron. It's called a gamma motor neuron. Let me use a different color. So I'm going to do gamma motor neurons in blue. And these guys innervate the ends of the intrafusal muscle fibers. There's a gamma motor neuron going to one end. There's a gamma motor neuron going to the other end of an intrafusal muscle fiber. Okay? So, if this little stringy thing here is an intrafusal muscle fiber, what I've been telling you is normally when a muscle length increases because we're stretching it, these intrafusal muscle fibers also get stretched, and that activates 1A receptors, and yay, we can tell the CNS that the muscle just increased its length. Follow me? But the problem is, when a muscle contracts, it gets shortened. So look what happens to the intrafusal muscle fibers. They get relaxed, and my 1A sensory fibers couldn't tell the CNS is what is going on. But if I take a gamma motor neuron on one end and a gamma motor neuron on the other end, and I contract an intrafusal muscle fiber, now it's taut, even as the extrafusal muscle fibers are shortening. Are you following me? So if we simultaneously contract a muscle with alpha motor neurons, shortening it, and we contract both ends of the intrafusal muscle fibers with gamma motor neurons, now the 1A sensory fibers linked to these intrafusal fibers, they're capable of signaling to the CNS regardless of whether the muscle is shortening or lengthening. That's how we solve the problem. So we solve the problem with having a whole second class of motor neurons. There's not as many of them. They're called gamma motor neurons, and they're innervating intrafusal muscle fibers. Okay. And you can see here, if we didn't have gamma motor neuron fibers, okay, when a muscle contracts and shortens, my 1A fibers go silent. They can't respond. They're not capable. So we're losing all that information. But if we now contract our intrafusal muscle fibers with gamma motor neurons, now during contraction of the large skeletal muscle, the 1A response, it just faithfully signals that, So even if the muscle is stretching or shortening, it's capable of signaling that. Do you see that? So that's thanks to what we call gamma motor neurons. Two kinds of motor neurons, okay, involved. All right. So now, 30 minutes. Clearly, you know, in a review session, you know, we're not going to go over every PowerPoint slide. What I want to do is, what I decided to do is, I'm just going to touch on a few things that I bet you were confused about. Maybe because I didn't teach it well, or just because they're inherently confusing, so that maybe you'll be less confused for Tuesday, right? So the first thing is, what about that retinal circuitry? Okay. So remember, big picture. Oh no, this is the wrong one. Sorry. So, let's just review a few interesting things. Here's the lens. Someone can make a lot of money if you want to go into engineering, just like make a better mouse, make a better interface with mouse to keyboard, to computer, so we can do this shit, pardon my French, better for you. Okay. Here's the lens. Okay, so light's going this way. So remember the first interesting thing. There's 3 neurons in the basic retinal circuit. And to get to the photoreceptors, this light has to go through all those neurons. So that's just weird, right? Although the fovea, right, most of them are pushed to the side, so that kind of solves the problem. But so the photoreceptors, the rods and cones are back here. Then we have what are called bipolar cells. And the output cells of the retina are right here. These are called retinal ganglion cells. And that's kind of one of the interesting things, right? And weird things about the retina. Light has to go through all these other cells before it can reach the photoreceptors where sensory transduction takes place. So you can lose information. Luckily in the fovea, right, that specialized area of the retina for high acuity vision, the bipolar cells and retinal ganglion cells are pushed to the side, you know what I mean? So light has direct access to the photoreceptors. So first, that's one of the weird things about our eyeball. Okay, second weird thing. Does anyone remember, are photoreceptors more active in light or in the dark? In the dark. Totally opposite what you think. That's totally counterintuitive. Why would you have the sensory receptor that responds to light be less active in response to light? That's just dumb. But that's what we see. So that's the second weird thing. Photoreceptors are active at night, and they're less active in response to light, okay? What do they release? Glutamate. Remember, those were cyclic GMP-gated channels. Cyclic GMP gates those channels open in the dark. And in the light, actually, cyclic GMP is degraded, enzymatically degraded, those channels close, and photoreceptors go quiet. Okay. So this glutamate release is in the dark. And if we shine light on a photoreceptor, actually less glutamate is released. Okay, third weird thing about the retina, okay? Anyone remember? Okay, let's just step back. Let me ask you a question. What's the major excitatory neurotransmitter of the brain? Glutamate. Okay. So when glutamate's released, and remember at axon, we talked about two kinds of receptors, right? It acts on NMDA receptors. This is a postsynaptic cell. It acts on NMDA receptors. And it acts on AMPA receptors. These are both excitatory. Well, remember all the weird things about NMDA receptor. It's like voltage sensitive, and it requires magnesium, and actually, not requires magnesium. Magnesium blocks it. It requires glycine. It's just weird, right? Okay. But this whole story, I told you, the first unit, glutamate's excitatory, glutamate's always excitatory, glutamate's, oh wait, I lied. Okay. In the retina, okay, actually there's two kinds of glutamate receptors. One of them is excitatory, as you'd expect, and one of them is inhibitory. Right? How could that be? The inhibitory glutamate receptor is metabotropic, and we know metabotropic receptors are hooked up to a G protein, and some G proteins are excitatory and some are inhibitory. So all you got to do is like magically make glutamate inhibitory, is hook it up to an inhibitory G protein receptor, and now you just magically made glutamate be inhibitory. Follow me? So in the retina, it's just weird. In one pathway, we have like what you'd expect, receptors that get activated by glutamate, but in this other pathway, like you wouldn't expect, there's this weird metabotropic glutamate receptor, and it's inhibitory. All right, so now let's look at circuitry. I'll probably confuse some of you. So three weird things, right, about the retina, at least. Fourth weird thing. This is a fun way to review. Anyone remember a fourth weird thing about the retina that we talked about? There's all these blood vessels on the surface of the retina. Light's going right into that too, and so that we shouldn't see anything where those blood vessels are. When we look out at the world, there should just be like this network of like spider webby blood vessels interfering with what we see. None of us experience that, so the brain is filling that information in, okay? And then also the optic nerve, where retinal ganglion cell axons leave the retina. There aren't any photoreceptors there, so that's a blind spot that we all have, and yet we don't see a blind spot, do we? We're filling that in too, so that's another weird thing. Okay, okay. Here's this dark current, right? Cyclic GMP gated channels are open in the dark. So photoreceptors release glutamate in the dark. Bizarre. Okay. It involves rhodopsin. Okay. Now, this is what I want to review. This is a photoreceptor and talking to two bipolar cells. One of these bipolar cells we call on-center, and one of them we call off-center. Remember, we already started to see really cool kind of like receptive field phenomena in the retina, where it's like, it's not just a circular region that if light shines on it, you activate the cell, but actually, right, there was like a center region that might be excitatory, and an outer region that might be inhibitory, so we have on and off cells. That's cool. So anyways, we have on-center bipolar cells and off-center bipolar cells. And turns out that these on-center bipolar cells, their response to glutamate is inhibition. That's weird. And these off-center bipolar cells, their response to glutamate is excitation. So the big picture here is, going all the way back to this theme from the first unit. It's the same neurotransmitter, but it can have completely opposite effects because there's two different kinds of glutamate receptors here. This glutamate receptor here is inhibitory. This glutamate receptor here is excitatory. Okay? So now, let's think about that. So, if this is an example of a receptive field in the retina, we call it an on-center receptive field. So if we shine light at the center, okay, we get activation, and then out here we get the opposite. So the question is like, why? If I shine light in the center, that light's hitting this photoreceptor. Now what happens to glutamate release from this photoreceptor? It reduces it, right? Because light stops glutamate release from photoreceptors, which is counterintuitive. So we have less glutamate being released. Okay. Remember, I just told you, again, on-center cells, in response to glutamate, they actually hyperpolarize. They get inhibited. There's a metabotropic G protein expressed by these guys, right? So glutamate weirdly inhibits this guy. So think about it. If I take away glutamate, I'm taking away that brake. So this guy now is super happy all of a sudden, like, oh God, I'm not inhibited. So this guy's actually excited. Okay? Even though less glutamate's released. You follow me? If this guy's excited, we just excited the output of the retina, the retinal ganglion cell. So that's why shining light on this photoreceptor leads to activity in this pathway. Got it? Now, this off-center bipolar cell, I just told you, these guys are excited by glutamate normally. So if there's less glutamate being released, these guys aren't excited anymore. They're actually inhibited. So in this off-center pathway, I don't have activity. So that's why I get this center surround phenomenon. Follow me? And you could see how we could use that to start to discriminate lines. And we talked a lot about that, how you can discriminate an area of dark from an area of light, which we call a line, right? Okay. But so, you know, okay. Let's do it in this case. What if light is shining in the surround for this particular receptive field setup? So now, in the center, it's dark. What happens to photoreceptors in the dark? They release glutamate. And if we're releasing glutamate, glutamate inhibits on-center bipolar cells because it's a metabotropic glutamate receptor. So this guy's inhibited, so this retinal ganglion cell is inhibited. So you see it? So now in the center, there's no activity. This is in the dark. And it's releasing glutamate. Off-center bipolar cells are excited by glutamate. That's what we expect glutamate to do. So this guy, it excites its retinal ganglion cell. So in the off-center area, now we have activity. Okay. Now, let's go back. Last comment. Okay. I want to get to a few other reviews. I just want to say, how could we even make this ability to differentiate darkness from lightness, which we call line discrimination? How could we even enhance that? You know, I showed you evidence that we actually see more contrast than actually exists in the world because of our retinal circuitry. Here's how we can do it. Remember, if we shine light in the center, less glutamate's released. If less glutamate's released, on-center bipolar cells are actually active. Normally they're inhibited. So blah, 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 a lot of activity here. If less glutamate's released over here, off-center cells are inhibited. So already we got discrimination, right? Activity, no activity. Got it? But, okay, in the surround of this receptive field, it's dark. So if there's a photoreceptor linked to the surround, it's actually releasing a lot of glutamate. And it's activating a horizontal cell. Remember like these little accessory cells in the retina I talked about? These guys are inhibitory. See what I'm saying? So this guy right here, this photoreceptor, it's releasing glutamate in the dark. It's exciting a horizontal cell. What do cells do when they're excited? They release their neurotransmitter. What's the neurotransmitter that horizontal cells release? GABA. It's inhibitory. And so now, it's a second reason this pathway, there's no activity. So because this horizontal cell is releasing GABA, it's like over here, there's less activity. You see what I'm saying? So we just increase the contrast even more. That's why we see more contrast than actually exists in the world. This is a neural circuit that enhances contrast. It's actually designed to make us see edges, black, white, even more contrast than exists in what the world is giving us. Do you see that? Just the way this circuit is designed. All right? Okay. I want to draw for you. Two pathways. This goes back to the first lecture in this unit. We spent a few days talking about the somatosensory system. I'm talking about like a pathway for pain and temperature, and a pathway for touch, vibration, things like pressure. Okay. So, and I feel like maybe I didn't do a good enough job. I think we talked a lot about the physiology, but we need to know a little bit about the anatomy of those two pathways. So I want to do that. So the touch, pressure, vibration pathway, we call that touch, pressure, vibration pathway. We call that dorsal column, medial limbiskis pathway. It's also crossed. So, okay, this is a 3-neuron pathway. So, let's do an example where we touch something with our left finger. Okay? So, I feel the pressure, or I feel vibration, or something like that. Okay? So, here's my left finger, and I just touched something. So, there's a mechanoreceptor, right? Like a pituitary corpuscle or vertical disc. It just got activated. And its cell body is in the dorsal ganglion. And what's activating this pathway? Mechanoreceptors, proprioceptors, things like that. There's also proprioception pathway. Let's say it was like a mechanoreceptor that got activated by that finger. Okay, so first neuron cell body is in the dorsal ganglion, and we come into the spinal cord. And there's that butterfly-shaped gray matter, dorsal horn, ventral horn. And what's interesting is we don't do anything in the spinal cord. Now we just go up the spinal cord. This is the midline. Go up the spinal cord until we get to the brainstem, spinal cord. Brainstem. And the bottom of the brainstem we call the medulla, right? So in the bottom of the brainstem and the medulla, we synapse on the second neuron. We haven't crossed the midline yet. Okay. So that's neuron number 2 in the medulla. Neuron number 1 in the dorsal ganglion. Neuron number 2 in the medulla. Neuron number 2 now sends an axon to the opposite side of the brainstem. Now we're going up the opposite side of the brainstem, and now we're going to go, right above the brainstem, we have the thalamus, the sensory relay station. And we're going to synapse there. That's neuron number 3. The nucleus is called the ventral lateral nucleus of thalamus. And now these guys, these third order neurons in the thalamus, they're going to project up to, these are the 2 hemispheres of the brain. So it's like the left side. This is the left hemisphere. This is the right. And they're going to project to what's called primary somatosensory sensory cortex. And when that information gets there, now you're conscious that you just touched something. Okay? So let's do another example like we did for cortical spinal tract, like if you had a stroke. So this left-sided touch information is going to the right side of the brain. So say I have a stroke on the right side of the brain, I might lose the ability to sense stuff on the left side of my body. Right? I have weird sensory phenomena, things like that. Right? I have anesthesia, whatever. Does that make sense? Okay. Now let's do. Okay. Now let's do the other one. I'll pick this one up. So now we have the second somatosensory tract. This is for pain and temperature. And this one's called spinal thalamic tract. And we'll do it again. We're starting on the left side, but now we're not touching something, or maybe we are, but now we're touching something that burned us, or a mosquito just bit us on the finger. Okay, so we're starting out here on the left side. Finger. And now it's the trigger is a nociceptor or a thermoreceptor. And cell body is in a dorsal ganglion. And now we're coming into the spinal cord. Something different happens. So we come in and we synapse in the dorsal horn. So this is neuron 1, and then the dorsal ganglion. Neuron number 2 is actually in the spinal cord. So this is different from here. Remember, neuron number 2 here is in the medulla, it's in the brainstem. Here, neuron number 2 is in the dorsal horn and the spinal cord. These guys then, these second order neurons, send an axon that crosses to the opposite side of the spinal cord, and they're going to ascend in the white matter down here. Okay. Now they're going up the spinal cord, opposite side, and they go all the way up the spinal cord, and we're going to go through the brainstem. And so we're traveling up there, and we're going to the thalamus again. Same nucleus. Ventral, posterior, lateral nucleus of thalamus. And third order neurons here are going to go to the same region of the right hemisphere. Called primary somatosensory cortex. Okay? So this tract also has 3 neurons, but it's crossing in the spinal cord. But same thing, like if you had a stroke involving the right hemisphere, you might lose the ability to sense pain on the left side of the body. Or sense temperature. There's really, I should have gone into this, but there's really rare cases, human cases of people who don't feel pain. Genetic condition where this pathway is not working. And you might think that's a great thing, like, you're impervious to pain. What a great superpower to have. But it turns out, think about the problems. Think about what that system's designed to do. prevent our hand that touched the hot tea kettle from burning too much. But if you don't sense it, then you're just going to continue talking to someone while your hand burns into a crisp. So they had these few cases just having major problems because they can't sense anything painful, any hot stimuli, whatever. Okay. So now let me put these pathways together with cortical spinal tract to end, kind of tie it into Tuesday. If we look at the brain from the side, so here's a human brain from the side. Here's the cerebellum back here, brain stem, spinal cord. This is temporal lobe, frontal lobe, parietal lobe, occipital lobe. Okay, just a little bit of anatomy we need to know. Now, if I take a little groove right here, I'm going to call it the central sulcus. Okay, so the little strip of cortex right in front of the central sulcus, remember we call that primary motor cortex. That was Tuesday's discussion. And the little strip of cortex just behind the central sulcus, that's primary somatosensory cortex. That's where spinal thalamic tract is going. That's where dorsal column medial limbiskis tract is going. Okay, they're going to primary somatosensory cortex. That's where they're located. The central sulcus divides frontal lobe from parietal lobe. Just tying in some other lectures from this unit. Remember cranial nerve 8, auditory vestibular system. The auditory part's heading here to the top of the temporal lobe, primary auditory cortex. My visual information is heading to the occipital lobe, primary visual cortex. We also talked about, if you took your fingers and you put them in this little sulcus here, separating frontal, parietal from temporal lobe, is called lateral sulcus. And if I put my fingers in the lateral sulcus and pull the temporal lobe down, imagine that, okay? I pull the temporal lobe down. Remember how there's a fifth lobe in the brain called the insular cortex? That's the location for gustatory cortex, right? Where taste information goes. So anyways, but that's this primary somatosensory cortex just behind the central sulcus. That's where spinothalamic tract, pain and temperature is going. That's where dorsal column medial limbiskis tract touch is going. Okay, and both of these, primary somatosensory cortex, primary motor cortex, there's a map of the body there. It's not an equal map. The map is biased towards the hand, towards the head, towards the face, and we have topographic maps. Okay, so I would be able to draw the cortical spinal tract, the dorsal column medial limbiskis tract, the spinal thalamic tract, some basic retinal circuitry. I'll give you more study guide information later tonight when I finish the study guide. Okay.